in really fundamental ways. So they differ in the amount of additives that are added to them. They differ in what they're used for, the sizes, either in the size that they are produced as or what they end up as once they're in the environment. The morphology of the plastic, whether it's a fiber or a sphere or a film, colors, and then their ability to have toxins that are already present in the environment attached to them also depends. So when we're thinking about plastic, it actually is this really diverse suite of materials that we have to be considering. And at this point, for the most part, policy hasn't considered it that way. So all these different types come from different sources, and so the route into the environment is also different. So something like plastic microfibers enters the environment through your washing machine, where it goes to the wastewater treatment plant, is not filtered out, and ends up in the environment. Whereas some of these other things are the result of either plastic mismanagement or littering. Tire dust has recently been identified as being extremely toxic to fish. That just comes from wear and tear, driving down the road. So there are a really large variety of sources and routes into the environment. And this also provides an opportunity to provide more directed management of plastic. So when we think about what it is, a lot of it starts out as macroplastic. So these are some figures that have made the news where on the left is one of these ghost nets. So these really massive polypropylene fishing nets that are just floating in the ocean. Or on the right are still macroplastics. This is an albatross, and you can see the amount of plastic that was ingested by the bird. Another one that gets a lot of attention are plastic bags that turtles mistake for jellyfish. But when we think about this ocean garbage patch, it's really more like this plastic soup that has both macro size, so big pieces, and microplastic floating around in it. So microplastics are less than 5 millimeters. There's also nanoplastic, which is getting down into the micron range. So the plastics in the environment span this whole range of sizes. And with the microplastics, again, some start as these very small particles and are actually produced that way. So nurdles are the kind of pre-production plastic that then are molded into specific compounds. Whereas the secondary plastics are ones that break down in the environment. So this would be, for example, a plastic bag breaking down into smaller and smaller particles in the environment. It's classified into a variety of categories: films, fibers, foams, pellets, fragments. And when we try to identify plastic that we find in the environment, we try to break it up into these different categories because it gives us a better idea of the source of the material. And these sources are very diverse. But we have a pretty big gap in understanding of what happens with plastic breaking down in the environment or breaking down during the use phase. So I mentioned before this tea bag example where it wasn't considered in the design phase that these will actually break down in hot water and then enter the environment as you're brewing the tea. So we really need a better identification of both the sources and then the pathways that they are entering the environment. Okay, so what we wanted to talk about was the Great Lakes and plastic in the Great Lakes, which is getting increasing attention, and that's true with freshwater systems in general. To date, though, the bulk of plastic pollution research is still focused on the oceans, but we're trying to move upstream and look at lakes. So we're mostly focused on Lake Ontario. With about 8 or 9 years ago, the question started to be posed about whether or not there was a Great Lakes garbage patch, similar to what we find in the oceans. And so there was a group of scientists that tried to go out, this is SUNY Fredonia and also groups up in Michigan, to try to figure this out. And so They took trawl studies through a couple of the Great Lakes. They found plastics in all of the lakes. And at that point, the main focus was on microbeads. And a lot of that work actually led to the ban on microbeads in personal care products because they found so many microbeads floating in the water. The work since then has shown that the plastics are pretty ubiquitous in all different types of Great Lakes environments. They're found in the sediments, where the work has identified that they've been building up since about the 60s or 70s in the environment, in the sediment. So it's a little bit of kind of like archaeology of sediments. They're found in rivers, both in the water and in the sediment. They're found near shore, they're found offshore. And one of the interesting things about looking at it spatially is that the type of plastic you find in these different environments varies. And a lot of that has to do with both the morphology and then the density of the plastic. Where the denser the material, the more it tends to be found in river sediments and near shore. And the further you go offshore, you find less of that in the sediment and more is still in the water column, particularly the less dense materials, things like the expanded polystyrene. It's also on our beaches. This is some data from the New York State beach cleanups. This is 2018, where they found 40,000 miscellaneous plastic pieces on the beach. And so those are just the ones that you can't identify. But if you look at this list of material that they're finding, almost all of them are some type of plastic. So the things that we're interested in are what happens once the plastic enters the environment, and how does that vary both spatially and temporally? So some of the things that we're trying to consider, the first one is transport, and so I'll be talking about Matt Hoffman's work on this. And then we're interested in what happens over time. So the material enters the environment, it's acted on by the environment in which it lands, whether that's weathering processes, so UV light, the turbulence in the water. It may absorb different kind of chemicals that are in the environment, so the toxicity may change. I'm interested in the toxicity of the compounds and how it affects ecosystem dynamics. And then we can look at how the materials may move up the food chain. So looking at the plastic effects, with these larger pieces of plastic, it's a lot easier to link the plastic to the death or toxicity in the environment. These are a couple of examples where a turtle was found with this huge amount of plastic in its stomach. Another is a whale that had 48 pounds of plastic in its stomach. So these are pretty obvious, where the plastic's consumed and it's having a toxic effect. With the microplastics, it's a lot harder to figure this out. There's been a huge variety of studies on ingestion of microplastics, primarily in invertebrates and primarily in marine systems. And the results are everything from reduction in feeding to neurological effects. But there's still a whole lot that we don't know about the impacts on ecosystems. There's a lot of potential for just straight up chemical effects. And some of this is, remember that list of additives that I mentioned of what is incorporated into plastic during production, but then also the absorption of different kinds of toxins while it's in the environment. And so these may actually increase the toxicity the more time the plastic spends in the environment. And the plastic may act as a vector for moving toxins around. So there's a lot of gaps in what we know in freshwater systems, and some of the gaps that we're trying to work on in our group at RIT is to look at the temporal and spatial variability in the sources, looking at the different transport pathways ultimately to the lake, looking at quantifying and characterizing the microplastic. So how much is it and what kind is it? And then what happens to it while it's en route from wherever it enters the environment into its end of life, where it may be buried and kind of taken out of the ecosystem. in order to be able to get at some of these more either polymer specific or pathway specific impacts on the ecosystem. The ultimate goal is then to be able to develop more targeted source reduction and removal recommendations that take into account both pathway and the type of plastic that it is. So RIT first got involved in this problem when Matt, who unfortunately isn't here, but he's an applied mathematician and he started to get interested in this problem. His background is in numerical weather prediction and ocean forecasting, and he started to think about plastic, trying to answer questions about how much is there and where is it in the lakes. Some of this was derived from research where we had ideas about how much plastic was out there, but actually doing this kind of sampling is expensive. It's really time consuming and it's super tedious. Um, so by applying models, Matt was able to get at a more complete spatial and temporal resolution of where the plastic was expected to be. And with Eric Hittinger, who's in public policy at RIT, they did some estimates of inputs into the Great Lakes. So the estimates range a lot depending on the assumptions you need to use about where it's produced and how it's managed. But about 10,000 tons of plastic enters the lakes every year. And then they were able to compare that to what was found in some of these empirical studies. So people that have gone out and actually measured how much plastic's in the water column. And there's a really, really big discrepancy. So the model suggests that we should find a lot more plastic in the lake than we actually find just floating in the water. So Matt started to figure out what some of these transport pathways in the lakes are likely to be, and keeping in mind that there are lots and lots of different types of pathways that the plastic can be following. And initially started looking at kind of these 2D transport processes. So the plastic comes in and it's transported around the surface of the lake. So he used these models, which it looks like may not play in Google Meets. It works in Zoom. Okay, so we won't be able to get this to play, but he used a 2D model that looked at the transport of plastics between lakes and within lakes with using these estimates based on human population. And a lot of what this showed is that there was not really an accumulation anywhere except for close to the shorelines, and that out in the middle it was actually more diffuse. Um, so there's really no evidence from this, from the garbage, that there would be garbage patches with much more accumulation near the shore. And when the model is compared to actual empirical data, this is for Lake Erie, it actually fits pretty well. So there's a pretty decent fit of the model predictions to the data. But there's still this question of where's all the plastic going? And so this is where Matt started talking with me about it, because I'm a benthic ecologist, and he was thinking that probably a lot of it ends up on the bottom. So we wrote a little paper to go into the conversation, making this hypothesis that a lot of it ends up on the bottom, but that we need to get a better idea of where it is and the sources and transport pathways, and then also the fate and impact of the particles and chemicals. So at that point, we put together a larger group of people. So now we have a larger interdisciplinary research team on this. So this is the type of problem that requires lots of different types of people to work on it. So this is our COVID research team, where we were not in the lab, but in our backyards doing research. And so we have Nathan on the left, who's our chemist on the project. We'll be talking in a minute. And then Matt, me, and then Stephen Day, who's a mechanical engineer, who's helping with some of the fluid mechanics of the problem. And then Andre Hudson, who's a molecular biologist and biochemist. And so now we have a pretty substantial research cluster trying to address the plastic problem in the Great Lakes. So with this new group, and we now have some funding from New York Sea Grant to look at it, Matt's PhD student, Juliet, has now moved from having a 2D model to a 3D model. And so instead of just looking at the lateral transport of the plastic, she's incorporating the sinking velocity. And she's been able to do this on a polymer specific basis using primarily the density of the particles and finding that much of it's sinking. And so this is in contrast to seawater, which is more dense than freshwater. That we find that in freshwater, the plastics, many of which have densities quite close to the density of fresh water, are actually sinking. But there's a lot about this that we still don't understand in terms of how much is ending up on the beach, how much is sedimented, how much of it may sink a little bit as it gets fouled by organisms and other contaminants, but then defoul and come back up. What happens as it gets smaller and smaller? Is it ingested? Do the organisms actually play a role in the bioturbation of it, so moving it around? So by incorporating these new vertical processes, these are the ones that are boxed in red here. Matt and Juliet were looking at the turbulent mixing, so how particles may move up and down based on the density, but then also on the water currents, looking at deposition to the sediment. And they did this for 9 different types of polymers. So this is a figure showing these 9, they're the ones boxed in red, that differ in sources. They differ somewhat in the sizes that they're coming into the environment as, and they differ in the density. So some of these have a positive sinking velocity, which means that they float, and the other ones have negative sinking velocities, but the sinking velocity will depend on the density of the material. These are the ones that are most often found in the Great Lakes. So they ran the model for about 6 months, and then in the water column, they found, which is not surprising, that these 3 that are positively buoyant are mostly found in the surface waters, and that they do get mixed a little bit below the surface, depending on the turbulence in the area, but most of them are still up at the surface. Whereas with the denser plastics, a lot of these, so for example, PVC is quite dense, that ends up on the bottom. And that the concentration of these are kind of at the ends of the basin, especially this western basin in Lake Erie. So using some mass estimates, so looking at 2 different sources that have mass estimates with concentrations in the environment and average particle mass, they are able to get at how many particles may be out into the environment and able to model then the deposition rate. And they estimated that in Lake Erie, the deposition of plastic is about 170 tons of plastic every 6 months ends up on the bottom of the lake. So that's starting to explain a little bit about where the plastic is going, that a lot of it is probably ending up on the bottom. So this is part of where we were coming into the problem with trying to look at it from a more interdisciplinary perspective is then we know that it's coming into the environment. We know that some of it is sinking, but how does it change while it's in the environment? And then how do these changes impact the ecosystem effects? So this is the suite of plastics we've been looking at so far. We have polystyrene, polyethylene terephthalate, and high-density polyethylene, which make up your classic water bottle and cap. Polyethylene terephthalate in a fiber form is fleece. SPR is crumb rubber, so this is what would come off of tires, but it's often recycled and used on things like turf fields. And then PVC, or polyvinyl chloride, which is common in construction materials. So we ask these questions about the changes vary depending on where the plastic is, so where it enters the environment, or on the depth within the water body. So we've set up an experiment over the summer where we deployed all of these different kinds of plastics, both in particle form and in what we are calling swatch form, so little pieces of plastic, at 5 different sites in our region. So everything from a rural pond down near Naples, New York, to one of the Finger Lakes and Kinesis Lake, a stormwater pond on RIT's campus, uh, the Tate Preserve Quarry Pond, which is near Ellison Park. It's RIT's new research site, and then up actually in Lake Ontario. We built these rafts and attached plastic to it. I like to joke that this looks like the junk art that my kids brought home from school when they were younger. And then they were deployed both at the surface and then this place. Also, they can see down at the bottom of this pond, we're also deploying them at the bottom. So these were set out in at the beginning of July at these locations, and then we're sampling them. So far, we've sampled after 1 month and after 4 months. We've got they're still out in the environment over the winter, and we'll be sampling again next summer to get a roughly 12-month sampling point to look at changes in toxicity, chemistry, biofouling characterization, and the settling velocity. So we'll right now, we're going to just present some preliminary data because we're still working through a lot of these data. Um, this shows some of the students working on our team, well masked and socially distant. And this is at the JLAP pond, and this is Lake Ontario. And then these are the samples collected after just 1 month in the ponds. So you can see this really big difference. This is JLAT. The plastics are really fouled. The ones on the bottom, it's very anoxic. Whereas in Lake Ontario, the ones from the surface are moderately fouled, but the ones on the bottom are fouled much more. There's actually a lot of algal growth that we got on the plastics from the bottom of Lake Ontario. So my student Christina has been looking at the toxicity of these particles. And she has completed now the toxicity experiments in these chronic exposure assays for plastics in their pristine form and then where they've been aged in the environment for 1 month. She's focusing on the difference between the stormwater pond and Lake Ontario. So our model organism here is Lumbriculus variegatus, which is a common benthic invertebrate, particularly in of impacted ecosystems. And she uses artificial sediments and artificial fresh water, so it's consistent across all of the treatments, and then runs the experiment for 28 days with and without plastics, and then she looks at a variety of different factors. Right now, I'm just going to present the population size. So she found that all of these plastics are relatively toxic. So this is the population size over the whole course of the experiment from the end. So we started with 15 organisms in each jar. In our control with no plastics, the population roughly doubles over that time point. But with the plastics, you don't see much growth in the population size. One thing I'm not showing, but that we actually see is that the individual weight of the organisms actually increases and relative to the control because the organisms, when they're stressed, tend not to reproduce, but they just increase a little bit in size. She'll also be looking at how much plastic they're actually ingesting that may be leading to that increase in weight. Then if we look at what happens to these aged plastics, so this is Lake Ontario and this is the stormwater pond, over this just 1 month period of the plastic being in the environment, the Lake Ontario plastic is almost the same as it was. It is a pristine plastic. In some cases, it's maybe a little bit less toxic, but about the same amount of toxicity, a little bit more toxic. But the stormwater pond, really interestingly, the plastics in the stormwater pond become less toxic over time. And so to the point that they're almost similarly toxic to the control. And these are just the surface samples, but the data from the benthic samples are really similar. So we see quite a big difference between different environments in what happens as far as the toxicity of the samples. We also run some preliminary samples looking at our COVID litter and find that these are also quite toxic, particularly the fibers that make up these the disposable masks. We're also now aging some of these in the environment, and we'll be doing some of the same studies on this. So one of the things this suggests is that there's going to be broader impacts of this. So the plastics are going to be influencing the diversity, which then increases the impact on ecosystem function, whether it's organism impacts or community level impacts. So ultimately, ecosystem services have been impacted. We have another whole suite of experiments looking at shifts in biogeochemistry and finding that there's also these kind of polymer-specific impacts on ecosystem functions. So the mechanisms of toxicity, when we start looking more closely, some of it is that the organisms are physically impacted, whether they're ingesting or being entangled by the plastics that are present in these environments. But that may not explain the physical piece of it, not explain why the toxicity varies across both space and time. So to do some explaining on what might be going on with this, I'm going to turn it over to Nathan.
Hello, thank you, Christy. So to go along with the idea of the toxicity, as well as also looking at in terms of the ultimate fate or sink of the plastics, we wanted to also look in terms of both the chemical changes as well as also the physical changes of these plastics. So what we're looking here is I'm going to talk just on one specific plastic. So I'm going to focus just on polystyrene and looking at both in terms of the two different types of styles of plastics we talked about in terms of being in more of a particle form or being more of in a swatch form. And if we look in here, this is looking at our photographs after 1 month of aged of polystyrene. In each of these pictures on the left-hand side is the image of the sample that was deployed at the surface. The ones on the right are the ones deployed in the benthos or in the down in the sediments at the bottom of the lakes. The first thing to note is you can see that the biofouling is very different between the locations. If we look in terms of JLOT here on the on RIT's campus, you can see up at the surface there's very large amount of biofouling, whereas in terms of it in the benthos, it is a very small amount. As Christy was saying, that in the benthos it's very anoxic, whereas there's a lot of growth that's at the surface in JLOT. If we look at Canisius Lake, we kind of see a difference. We can see that you're getting a fair amount of film growth on the surface in the benthos, whereas at the surface, we're getting more of this yellowing. So we're looking at, you can see that there's site-to-site variation in the extent of biofouling. In addition to this, we're looking at the settling velocities, how the settling velocity changes after aging, as well as also the microbial metagenomics of this the biofilm formed, as well as also the antibacterial resistance of the bacteria that is formed on these different samples. So this is looking at it in terms of after 1 month. You can see, like I said, that's both differences in particle versus swatch, but also in terms of different locations and surface versus benthos. As Christy was saying, we have recently done the 4-month sampling, and we have pictures of these as well. And so here is looking at the biofouling after 4 months in the environment. Couple things to note. One, especially if we look like JLOT, look at that sample from the surface. It is very, the biofilm now is, or biofouling is to a very large extent. And this is increased even from the first sample. If you note in Canisius Lake as well as also Lake Ontario, you see that we only having the benthos samples. In some of these, we had some loss of some samples. But something to note is that, like I said, we're seeing more biofouling as we go to longer time. And we're also seeing two different effects occurring. One is being this biofouling, this film being formed. We're also seeing the biodegradation. And for instance, if we look at like the samples from the pond, you can see this yellowing and this clouding occurring. This is indicative of like some oxidation occurring of these films as well. So we can think about in terms of both chemical changes along with these physical changes. The increasing of these biofilms will probably change their densities to think about in terms of being able to, that's why we're looking at the settling. Think about in terms of you increase its density, it's going to settle more. So even if you start out having a plastic that was less dense than water, that could eventually still settle out. And then, like I said, we're also looking in terms of what happening to the chemistry. So to also look at the morphology, we've also taken high magnification SEM, so scanning electron microscopy of these different samples as well. So again, we're still looking at the polystyrene, and for SEM, we're still in low magnification. We've taken much higher magnification as well. You see the same type of ideas. And this is really confirming the same spatial differences of the biofouling. When I say spatial, I'm talking about in terms of the Canisius versus Lake Ontario versus these ponds. That we can see the surfaces are, whether we think about in terms of having light biofouling, we look at like Canisius Lake, or in particular, like Lake Ontario, where you can see that not only do we see different amounts of material on the surfaces, but the construction of it is very different as well. If you think about in terms of location to location, which is probably going to have an effect on the settling as well as everything else. So this so far has looked at all of just the physical changes. But we also want to know in terms of chemically what's happening to the surfaces. So my students were also looking at doing spectroscopic analysis of these plastics before and after aging. And in all of the next 4 slides, you can see that the spectrum in orange, to reddish orange, is the control. So this is polystyrene before it is aged. In the blue, it's always going to be the sample that's at the surface. These are being swatches, and the black is being the benthos. I also have a picture of what polystyrene looks like. You can see we have this benzyl ring, as well as also this backbone. So we have a number of places where oxidation can occur. What we're mostly looking for is, are we seeing hydroxyl groups being formed, OH, as well as carbonyls, to really indicate about the degree of oxidation. So the oxidation is going to lead to greater hydrophilicity. So you get more water content under the surface, as well as also changing the binding potentials, whether you think about of chemical, or you can think about in terms of microbial as well. So if you look here, this is looking at Canisius Lake. We can see that we're getting this oxidation species observed in the benthos species. But we can definitely see that there is some changes in the spectra of both the surface and the benthos. If we move on to the next one being here, Lake Ontario, in Lake Ontario, we're really seeing no formation of groups, but we're definitely seeing formation of the carbonyl. And now this time it's being at the surface rather than in terms of the other one, which is being the benthos. If you really look at the surface samples, you see no differences between the pre-deployment and the spectra that we see after a month of aging. We still need to see like the T4 and the T9 or T12, depending on how long the last one's left out there, to really see about in terms of how this chemical is changing. So that's very preliminary, but we can also see that there is, down at the lower wave numbers, we're seeing some changes as well of the spectra. So we're definitely seeing surface changes of these. Moving on now to the pond. So this is that rural pond down near Naples. We're starting to see some actually very large changes. And if we look again, this is being more so at the surface than the benthos. And if we look at that picture there, you can see that the sample from the surface was very much yellowed, very much clouded. So really this oxidation, whereas the one down at the benthos was not as affected. And like I said, in terms of look at the spectra, it's almost identical to before being deployed. So this chemical changes are being very dependent location, both within the water column and also in terms of spatially where they're at. Finally, looking at JLOT, where we saw a large difference in the film being formed at the surface versus the benthos, is that if we look at the plastic itself, so the blue versus black and the orange, there is some subtle differences, but there isn't a large amount of difference. What I also have on here is looking at that biofilm itself. So that's the spectra that's in green. And what's different about the film from JLOT as the others is that this is a very robust film. There is a very strong adsorption of this film on the JLOT sample. To remove the film, I had to actually use isopropyl alcohol and really rub it off to really be able to get the spectra of just the plastic itself. And truthfully, this large absorption of this biofilm might be one reason why we're seeing less toxicity in the JLOT than we did in Ontario. Because if we have this film being formed, that we are now, the organisms might not be as exposed to the plastics once you have this film formed. So that might be some reason. And like I said, this is by far the strongest adsorptive film that we have observed from all these species. So that's kind of, like I said, we have a lot of preliminary. In addition, we're also looking at the additives. So we're looking to see about in terms of leaching. So we want to see about in terms of after being exposed, are there less additives in these different plastics? We have noticed that they are more brittle, but we're trying to dig into the chemical composition of the additives as well after exposure. And that's data is to come at a later time.
Okay, great, thank you. So we still have actually quite a lot of work coming up on this project and a lot of ideas about where to go with things. So what I wanted to just do a real quick overview of some of the future work that we're doing. A lot of this is some slides that Matt contributed, but we are all working together on some of the questions that are happening with this. One of the parts that I'm interested in and that we're also working with Stephen Day, who's a mechanical engineer on, is trying to understand the role of the benthos in the plastic fate. So this includes the organisms that live there. And some of our preliminary data suggests that once these particles get to the bottom, the organisms may actually act to further bury the plastic, kind of putting it out of circulation in the water column. And some of it is actually just physically moving it around. Others may be filter feeders that will ingest the plastic and then it's deposited on the sediment surface, kind of in a these pseudofeces. So there's actually some kind of reciprocal effects of organisms on plastic in addition to the plastic influence on the organisms. So getting better ideas of how does biofouling interact with the plastic to influence whether or not it sinks, and then what happens when it hits the bottom are some of these important ideas. We're also starting to move kind of further up the watershed to look at kind of spatially distinct sources of plastics because the different plastics have different fates in the environment. Understanding the route of transport to the environment will give us some idea of what the plastic is and how it's likely to have transformed during its pathway. This is a recent study that came out looking at the types of plastic found through different sources. So comparing a wastewater treatment plant where it's mostly fibers from clothing. The agriculture sources are kind of a mix. Stormwater is also a mix, but we see the influence of tire particles that we actually see in the lake are mostly the particles that are more buoyant because a lot of the denser particles settle out really quickly close to the source. So better understanding the spatially explicit sources will help to improve the model predictions a lot. Another one that Matt's been working on, and this is some pretty neat preliminary data, is trying to get at more spatially explicit estimates of macroplastic littering rates. And so he has data he got from Chelsea Rockman's lab at University of Toronto, where they do these litter audits, going out in the streets and actually picking up trash and figuring out where it came from. So the purple dots, the more dense areas of trash, and then he's comparing that to tweet density. So looking at where the most Twitter posts come from, which gives you some idea where the most people are. And you can see a really nice correlation between the activity of people and the location of the trash. So this is one way to perhaps get at litter rates and then the amount that's likely to end up in the environment. Some other work that we're just getting started on is looking at tributary data and trying to estimate based on weather data and flow data, how much loading is likely to come from different tributaries. This is also then being scaled against population density, but also needs to be verified with actual trash collection in the field. And we're working on some potential ways to be monitoring the macroplastic trash coming in through some of these different tributaries. Another approach that Matt and his student Juliet are taking is to look at where trash comes in and then following individual particles in through their models and seeing where they end up. So this top panel is where floating particles that enter from Toronto, which is the biggest population center on Lake Ontario, where they end up. And then they also are trying to go backwards. So they're saying, okay, here's all the particles that ended up at the beach at Sodus. Where did they come from? So this is then taking those particles backwards to figure out where the largest sources for specific end locations are. So this is work that's in progress now. So just to summarize, there's a lot of data needs still to define both what the watershed sources are and how they vary spatially. Looking at site-specific plastic accumulation and then understanding the fate and transformation based on where it's coming from and what happens to it en route. Ultimately, the hope is that some of this information that's more polymer specific and site specific can allow recommendations about behavior change and where the impacts are higher for both disposable plastics and durable plastics. Looking at kind of more diverse strategies to intercept the microplastic before it gets into the environment. And then also redesigning objects that have high impact with this idea of plastic shedding in mind to try to prevent shedding. So there's a lot of different people working on this project that we owe big thanks to, especially the huge number of students that have been working on this. And with that, we'll see if anybody has any questions.
Yeah, no, thank you. Nobody has posted any questions in the chat, so if anybody wants to post any, you can. Or still not seeing any. Oh. Carl, do you have any plans for citizen science teams, I think?
Citizen science teams, yes.
We've talked about that. And so to some extent, a lot of the beach data is actually citizen science data because people are going out and a lot of the beach cleanup data ends up getting documented and doing that. And then we also, it's actually part of what would be a new proposal would be implementing some citizen science more similar to what they're doing in Toronto with this kind of litter collection to try to get at things a little more spatially.
Okay. Another question with the plastic ban, bag ban in New York, do you expect to see fewer plastic film items in the lakes?
Thanks, Chris. Yes, I think so. It's, I would hope that that's the case. The bag ban just went into effect, so it'll probably be a little while. And films are a pretty common material that you find. One of the things about plastic films is that they also are the ones that break down the most rapidly, and so a lot of those particles get smaller and smaller and smaller, but they don't necessarily go away. So it may be that there's less new input, but the material that's there is going to persist for a while.
And is there any research into how turnover in the lakes impacts the toxicity?
Not that I know of, but that's a good question, and that's something that I think looking over time could potentially give some insight into that. A lot of the sites we're working at right now are very shallow, and a lot of that's mostly just because of access, and that they're not really stratified in terms of a persistent summer stratification. But what we saw with just this J-lot pond, where the bottom, even though it's really shallow, is really anoxic, the plastics are actually really well preserved where it's really anoxic. And so I think if you did see turnover and either resuspension with the turnover, with plastic going back up above the water column, above the thermocline, or just oxygen mixing down, you may see seasonal differences in the toxicity and the fate. So I think that's a really neat question.
Okay. And one last question. In regards to the fouling potential acting as a barrier between the living environment and the plastic pollution, have you seen any indication that fouling is accelerated the breakdown or otherwise chemically negating the toxicity of the plastic itself?
I would say with that, we need the data from the 4 months and the 9 or 12 months to really start to. It's definitely something we're interested in looking at. I think it'll be really interesting. My guess is truthfully with ones that are putting on these very strong, like the J-lot, I think we're going to see slower degradation. So in terms of that, it's one of the, if we think about in terms of one of the largest causes of degradation, with two of them, one is going to be bacterial, another one's going to be in terms of sun. So UV light does a lot of the degradation. So if you get a very strong adsorbed biofilm, you're going to see less degradation of these polymers, I would assume.
Okay.
Yeah, one of the interesting things we saw in Kinesis Lake was that a lot of the plastics are covered with zebra mussels. And where the zebra mussels are on the plastic and you pull them off, it actually looks perfect. So there's really nothing happening when you get a lot of biofouling with zebra mussels. So there's a lot of different types of organisms that foul these things. And I think that looking at some of that, organism specific information will be kind of neat. With the sediment and bioturbation, that's actually something that is on our future wish list. And some of that would be looking at how much you may expect in a site, and then you'd have to also go out and look at the organism density to get at that in a field scenario. But in the lab, we are doing some experiments looking at the phytoplastics when we use zebra mussels and different species of worms, and measuring whether the plastic is actually incorporated deeper into the sediment. And we do find that. I actually find that these little lumbriculid worms, the ones that we were using for the toxicity experiments, they seem to pull the plastic down into the sediment. And so we're looking at that more. So I think that that's something I'm really interested in, kind of finishing up those studies. We've got a great undergrad working on that, and so I'm excited to see what she's going to come up with in the spring. So yeah.
And then
Let's see.
last question on the list is there a common analysis that you could suggest area high schools and colleges and community colleges could work on simultaneously?
I think some of the getting at the the trash accumulation in different areas, and having kids go out and both pick up the trash and then quantify the trash, and being able to look at different sources in different areas, kind of crowdsourcing that information, I think would be really, really cool. And a way to not only get data, but also encourage the students in different areas to become more aware of the problem and more aware of that, and the materials that we're finding in the environment are materials that they've likely interacted with almost on a daily basis.
Okay.
And no, she's not allowed to eat the plant. She's feeling neglected.
Okay, well, thank you so much for presenting.
Yeah, thanks for inviting us.
And hopefully, we're still working on trying to figure out the conference and such. So keep your eye on your emails for that, and have a good rest of the day. Thank you.
Thank you all.
Yeah, thank
Thanks.
you. Bye.
Thanks, Susan.
Thanks, guys.
Thank you.